fungi are used for food. And when it comes to distribution and aggregation, the potential for incorporating mycelium-based packing materials, there's probability there. Fungi are already used to process and produce different types of fermented foods, which then, of course, requires some marketing before they can be purchased by consumers and ultimately prepared for consumption. The part that most interests me about this, the role that fungi play in food systems, is how they can be used to as a resource for waste recovery. And so that's what I'll talk a bit more about when we get to my thesis project. But to begin, I'd also like to just provide some kind of pretty basic background on the role of fungi in ecosystems and food systems. So currently, the primary role of fungi in our food systems is for food. And most of us are familiar with mushrooms, primarily the genera, the Basidiomycota and Ascomycota. Those are your mushroom-forming fungi, your morels and chanterelles. And down in the lower left corner, you'll see a photo of the classic white button or cremini or portobello, which is all Agaricus bisporus, your kind of common grocery store mushroom. But fungi also play a huge role in producing a lot of different types of fermented foods. And commonly referred to as yeasts, that's primarily the Saccharomyces species. And Saccharomyces cerevisiae is one of the more commonly used yeasts for fermentation of beer, wine, and the leavening of bread. So again, we're utilizing these fungi to produce these types of specialty foods that we know and love. Tempe, Rhizopus oligosporus is another example of a fermented food based on essentially a fungal fermentation. And then we have soy sauce. It's also fungal fermentation. Kombucha, the term SCOBY means it means a symbiotic community of bacteria and yeast. So that's kind of a community ecology take on fungi and the production of a food product. And then, of course, all of our aged cheeses, Penicillium being the primary species that get used for cheese fermentation. But there are all types of different species of fungi that are used for these specialty products. And I'd say we're still really just at the tip of the iceberg in understanding the potential of this vast kingdom for producing really interesting and tasty, delicious foods. And if you're really interested, you kind of get into the community college aspect, or community college, excuse me, community ecology aspect of things. And like cheese rinds, for example, are really rich and diverse ecosystems into themselves. The other thing I'll mention is the enzymes, fungal enzymes that are used for food processing. Fungi are used industrially to produce these different enzymes at very large scales for other different types of food. I put up the example of xylanase being one that's a fungal enzyme that's used for fruit juice clarification and also for beer clarification. So essentially, it's a fungal enzyme that industrial microbiologists produce at massive scales and goes into a lot of different processed products. So again, these are kind of the various ways that fungi hold space in our current food system. And returning, not only as food, but fungi are useful for a lot of different things. And although they've been used as food and medicine for centuries, more recently, their mycelium has been employed to create different types of materials, like biodegradable alternatives to building insulation and packaging materials you see in the upper right-hand corner. And there's a company based out of Troy, New York called Ecovative. And this is a photo from their website of these various different types of mycelium-based packing materials that are also biodegradable. So yeah, fungi are really useful. And again, I mentioned enzymes. This is the central photo is of your classic oyster mushroom, Pleurotus austriatus, a white rotter itself. And white rot fungi produce enzymes like most fungi produce enzymes, but they produce enzymes to break down these complex biomolecules that constitute plant structural proteins like cellulose and lignin. And these compounds have similar chemistries to different types of persistent organic pollutants in the environment, like petroleum or different pesticides or herbicides. So again, it's the fungal metabolism, the production of these enzymes and other types of metabolites that really make them super powerful for their role in decomposing different types of wastes. So yeah, when we think about restoring soil health, fungi could play a very central role in that aspect of our food system. This is a little bit of a messy depiction of the role of fungi in the carbon cycle, but through my master's work, I became just kind of fixated on the fundamentals of mass and energy, and essentially that we have these different pools of nutrients, elements like carbon and nitrogen and phosphorus, and they play these different roles in the environment. And when we find ourselves in a situation where we have, say, too much carbon in the atmosphere or too much nitrogen or phosphorus in our fresh waterways, that's really just a matter of matter out of place, or MOOP, as those Burning Man patrons would refer to it. But rather, I don't like to think of these things as pollution, but naturally occurring substances that human influence has essentially disrupted the natural cycling of. So when I've presented on this before, I've found that some folks have become confused about phosphorus as a pollutant. And it's not a pollutant, it's an element. It's essential for all life processes. And we should just think of it as something to redesign our flows to better create a more circular flow of these materials. So I threw this up here, the carbon cycle to begin with. And fungi, as I mentioned before, are primary decomposers in the environment. They are heterotrophic organisms, which means that they're not able to produce their own food, unlike plants, which are considered autotrophs. So they do produce their own food through photosynthesis. And they sequester carbon dioxide from the atmosphere and use the sun's energy and water, again, to sequester that carbon in their own tissues. And then their tissues are consumed by herbivores, who are then consumed by other consumers, predators, on and up the food chain. And ultimately, the dead leaf and plant matter and dead bodies of other organisms forms all of the material for which these primary decomposers, the fungi, can get their energy and reincorporate the mass that was once another organism into their own biomass. And they have a couple of different strategies for doing so. Some are obligate parasites, which means essentially they require a host. Some are weak parasites that require the host in some of their life cycle, but may have another stage of their life cycle where they're able to live somewhat independently. Then there's the symbionts, the mycorrhizal fungi, which partner with plants, who again are sequestering that carbon, producing sugars for energy. We all need sugar for energy, carbohydrates essentially. So those plants are sequestering that carbon as sugar in their own tissue. And with the mycorrhizal fungi, there's often a trading system that occurs where fungi are trading nitrogen, phosphorus, and other micronutrients in exchange for carbon that they can then incorporate in their own biomass. So that's a more symbiotic strategy, life strategy that certain types of fungi have. in terms of the nitrogen cycle, nitrogen is also an essential nutrient for life. It's found in all proteins and DNA, and it has it has both solid and gaseous phases. So it exists in the atmosphere. It's actually the dominant element in the atmosphere is nitrogen. And essentially, nitrogen enters into soil through nitrogen fixing bacteria that are present in the root nodules of plants. There's also some that fix it directly into the soil. But once that nitrogen is in that soil, it's in the form of nitrogen, or pardon me, it's in the form of the biomass of the bacteria themselves. So again, we have to think of ourselves as carbon storage, as nitrogen, phosphorus storage, and all the other elements that make up our tissues and run our processes. So we're all essentially, stored energy and elemental mass. So you think of, again, those dead bacteria or those dead plants and animals, they're attached to carbon, so they're organic. As a biogeochemistry nerd, when I hear the term organic, I don't think of the way that the food was made so much as I think of organic means attached to carbon. Inorganic means free of carbon. And it's inorganic forms of nitrogen and phosphorus that are bioavailable, as we say, for other organisms to then utilize in their various metabolic processes, like, say, the production of a protein or DNA. So I think it's just important to, again, recognize the role that fungi play as decomposers in the nitrogen cycle. And then I'll just also mention that nitrogen is commonly a limiting nutrient in terrestrial systems. And that means that it's one whose scarcity limits growth. And oftentimes in agriculture, farmers are applying nitrogen fertilizer to increase their crop yields. And that's because they have these plants have this kind of unlimited source of carbon in the form of carbon dioxide in the atmosphere, and an unlimited source of energy from the sun, but they need, again, nitrogen and phosphorus and other micronutrients, as we say, in smaller amounts. But nitrogen, because it's basically required by every living being, there's a lot of competition out there. So that's why in agricultural systems, we add a lot of nitrogen fertilizer to increase our plant yields, because there's often not enough stored in the soil. But when that nitrogen, say, runs off into aquatic systems, it can support the life of aquatic plants and kind of disrupt that natural equilibrium and become a problem from the human perspective. So I hope that that's all clear. I'm going to move on to the phosphorus cycle. And again, phosphorus is another one of these essential nutrients. It's used in the production of ATP for photosynthesis, in mammals for filtering waste in our kidneys and repairing tissue. It's what our bones and our teeth are largely made out of, is phosphate and other minerals. And that's how it primarily exists in nature, is as phosphate, or PO4. And it has a 3- charge, which a lot of cations or metals like calcium and iron and magnesium they have like a +3 or +2, so they're attracted to that phosphate. So phosphate is almost always either present in the environment as a mineral source, as a rock, or in solution dissolved in water. And the main thing I want to draw, the main point I want to make with presenting the phosphorus cycle here is that phosphorus has no natural gaseous phase. It's not recycled through the atmosphere. And so phosphate rock as a fertilizer, you hear, again, nitrogen fertilizer, phosphorus fertilizer, you go to the plant food store and you buy a bag of fertilizer and it has an NPK ratio on it. And that's the ratio of nitrogen to phosphorus to potassium. I'm not going to talk about potassium here, but N and P are the big ones because they're sort of the most crucial for these different metabolic processes. But again, to just return to the point that phosphorus does not have this gaseous phase and essentially only occurs in mineral form. So what that means is our sources of phosphate rock for fertilizer are finite and limited. So unlike nitrogen, where through the Haber-Bosch process, we can actually create nitrogen fertilizer by pulling nitrogen out of the atmosphere, we don't have a process for that with phosphorus. So we mine it from rock stores in what amount to very few locations on the planet where phosphate is economically mined. So finding alternative sources of phosphorus is really challenging, and developing processes that reclaim phosphorus in usable forms is really important to generating a more sustainable food system, because of course mining requires a lot of energy, and there's geopolitical consequences of the limited stores that occur only in a few places on the planet. And again, I'll just kind of like finish up with the fact that, yeah, phosphorus is also often a limiting nutrient in aquatic systems, which means when you have too much phosphorus, or I shouldn't say too much, but when phosphorus is out of balance in an aquatic system and you put more into freshwater systems, that results in a big bloom of life, often algae blooms. And many of you have probably heard about the negative effects of these algae blooms for water quality. And so that's why we get concerned about phosphate runoff from farms into our fresh water sources, where it could be considered a form of pollution. So to just kind of conclude, fungi in the environment, they are primary decomposers that break down these complex organic molecules into simpler forms, and they're big regulators of nutrient cycles. And they do this primarily through the production of these extracellular enzymes. So they secrete them out of their tissues, and that helps degrade these other organic compounds and freeze those nutrients from that carbon so that they're then available for biological processes. And that's exactly what we do in our guts when we eat food. Our gut flora and fauna essentially produce these different types of compounds, organic acids and enzymes, and break that carbon apart from those nutrients, and then we're able to use those nutrients to power our metabolism. So that's how life works, and that's why we care. So moving on to mushroom cultivation in a circular economy. I hope that most of you got a chance to read this fun paper from Grimm and Wuston. In applied microbiology and biotechnology, I really enjoyed this paper, and I really have come to think of a lot of our human processes in terms of how can we better achieve a more circular flow of mass and energy. And there'll always be some loss to some degree. It's hard to engineer a perfect process, and nature doesn't necessarily, have a perfect process, but it has a pretty darn good one for the most part. So in terms of thinking about mushroom cultivation, what goes into mushroom cultivation is from the start is raw biomass. So we need some material to start with to be our mushroom substrate, right? And that starts with agriculture and horticulture. And oftentimes we don't need to use the food that we would be growing crops for. We can use the byproduct. So if you grow a bunch of wheat, then you're left over with a lot of straw. So we can use that straw to become a mushroom substrate, where we can harvest or we can cultivate and then harvest some of these lignocellulosic loving fungi. So these white rotters that degrade these substances and produce fruits, mushroom fruits. So when we would do that, when we grow fungi, we're left over with not just the mushrooms, but also the spent mushroom substrate. And that has potential different uses as well. Depending on the species and the original substrate material, it might be a good animal feed, it could be a good compost, or it could be recycled into, say, an anaerobic digester or burned for bioenergy. So again, thinking about mushroom cultivation as a strategy, but also remembering that when we're cultivating mushrooms, we're producing mushrooms and also the residual byproduct, which is spent mushroom substrate. So thinking about how best to engineer that process so that we have additional uses going forward and we keep that cycle moving around. So now I'll go into my own research. I'll call it a case study. So this was the work I did for my master's thesis at the University of Vermont, and I've retitled it here as Integrating Mushroom Farming with Organics Recycling to Improve Water Quality in New England. And again, we've kind of touched on a few of these ways that fungi play a role in the current food system, but this portion of the talk is really, primarily for the resource and waste recovery, and then also for food production. So just going back to that food system elements diagram real quick. So a little bit of background. When I began my thesis, Vermont, which is where I live, had just passed this Act 148, an organics recycling law. And so essentially what that means is that no longer are we allowed in Vermont to throw away organic material into the garbage can. It has to go into some other process. And other states are doing this as well. On the left, you see essentially like it's a pie graph of our waste stream in Vermont. And then in response to this, what you see is that almost a third of the entire waste stream is food scraps and organic materials, which could be diverted away from the landfill. And this is largely, again, this policy is designed to reduce greenhouse gas emissions from landfills and to extract greater value from these organic residual materials. And so in response, the state adopted this food waste recovery hierarchy to help guide policy. And again, the advantages of this are increased recycling, reduced methane emissions, and then the opportunities to develop markets for reclaimed nutrients to better facilitate that circular nutrient economy. And while markets already exist for recycling paper products and other recyclables like glass and plastic, they're not always available, but markets for organics are less developed and present a greater management challenge. So as I said before, to guide this management, Vermont adopted this food recovery hierarchy, which essentially, aside from source reduction at the very top, results in end products that are destined for land application. So if you take these food scraps and organics and you compost them, let's say, I'll start with, you feed them to animals. You take food scraps, you feed them to chickens and pigs. What you wind up with is animals and animal poop. You've got to do something with that poop. It's going to go onto the landscape as a fertilizer product. The same with composting and anaerobic digestion. You produce some waste heat, biogas energy, but ultimately you're left over with compost or digestate. And those products ultimately are destined for land application. So some key take homes is that greater than 50% of the total waste stream can be diverted from landfills. This is, again, Vermont. About a third of that total waste is organic material, which can be recycled for energy production or nutrient recovery. There's similar laws being enacted or already enacted around the region, but the Vermont law was the most stringent. Anaerobic digestion, a quick overview. Anaerobic digestion is essentially fermentation in the absence of oxygen. And so what we have in Vermont and much of New England is largely a dairy-based food system. We've got a lot of dairy farms. You've probably heard of Ben & Jerry's and Cabot cheese. And those are two of our main export products and two of the biggest companies in Vermont. So we're largely a dairy state, although farmers are struggling with current milk prices. We still are primarily a dairy state. And as a result, we have a lot of dairy manure digesters on these farms. They're already in place. And so these farms or these digesters, essentially, they ferment manure, they produce biogas, which is then burned for biogas energy. And what's left over is an effluent product. But again, in terms of the food waste recovery hierarchy, anaerobic digestion is an attractive method because these facilities already exist throughout the state. There's 26 of them throughout New England and 17 in Vermont, and many of them are already accepting food waste. Again, to go back to the issue of nutrients and fresh water, we have a big phosphorus problem in our freshwater lake. Lake Champlain is the largest freshwater lake in New England, and it is making the news a lot lately because of these harmful algal blooms that are resulting in beach closures, reduction in property values. It's really affected the tourism industry, and it's been largely traced to agricultural runoff, phosphorus loading from agricultural runoff on dairy lands. So the big question is, how can we reduce phosphorus runoff and just alleviate the phosphorus load on a lot of these farms? So I began by constructing this diagram of this dairy farm cropland system, where the primary inputs are feed and bedding for cows. This goes into the husbandry of the cows. The cows produce manure and soiled bedding, in addition, of course, to the primary export product, which is milk. And so that manure and soiled bedding material becomes the primary feedstock for the anaerobic digester on the farm. And that is to produce, as I said before, biogas energy. Now, when we introduce organics recycling to this process, I'm sorry, I should go back and just walk us through the rest of the diagram before I get too far ahead of myself. So again, I said manure and soiled bedding, that's the primary feedstock for the digester. On the back end, that is separated into solid and liquid fractions through, most typically on these farms, a screw press. So that screw press like squeezes the leftover effluent and puts the water, the liquid effluent, into one place for storage, where it's ultimately applied as a fertilizer product for feed crops and pasture to grow some feed for cows. But again, that poses some eutrophication risk downstream. And I should have said this before when I had that slide up about runoff and all that green, gross algae, but that's what essentially eutrophication is a term for nutrient pollution in freshwater systems. And that's when you have essentially too much life, too much plant life, that, you know, these algae blooms wind up requiring a lot of oxygen while they grow. And so that deteriorates water quality for other organisms that maybe we appreciate more, like fish. But then to go back, so the solids are ultimately separated and dried or composted. Some are sold off farm as an amendment product, but often, again, recycled back into the digester as a bedding material for the cows. So that's one reuse of the bedding. But the big question is, okay, so this is a diagram of these human-mediated nutrient flows on a typical dairy farm with anaerobic digestion. But how will introducing new inputs of nutrients in the form of food wastes affect these farm nutrient flows? And the advantage of this is an increase in biogas production. So by bringing these food scraps and food processing residuals as a new feedstock for the anaerobic digester, we increase biogas and energy production, which can be recycled on farm or sold off farm and back into the system, again, capitalizing on a waste product. But how will that increase potentially affect these residuals? You know, there could be potential to exacerbate that eutrophication risk and freshwater pollution. So that was the main question that I was considering, was with this new organics recycling law that's steering food waste towards digesters, how can we generate a product that might capitalize on this increase in digester or digestate waste in a way that provides value for farmers and exports nutrients off of the farm. Well, the good news is white rot fungi to the rescue. As I said before, edible white rot fungi constitute a multitude of species which grow on a variety of agroforestry or lignocellulosic wastes. That stands for lignin and cellulose, these primary plant structural proteins. And this graphic on the right, I think, is really powerful. This is from the World Resources Institute. And what it suggests is that supplementing animal protein-based diets, especially beef, with mushroom protein could drastically reduce emissions, water, and land use. And that's, again, what it shows. You know, if we supplemented 30% of a beef burger with mushroom tissue, it'd be the equivalent to taking these 2.3 million cars off the road, conserving 2.6 million Americans' households worth of water, and reducing agricultural land demand by an area larger than Maryland. So I chose to work with Pleurotus austriatus to essentially be my test species. And this is your common oyster mushroom. It's the second most widely cultivated species worldwide, and it has a nutritional value comparable to eggs, milk, and meat. So it's high in protein. Cultivation on substrates supplemented with solid digestate had been done with digestate from corn silage, cattle dung, broiled chicken litter, etc. These other listed here. But there hadn't been any studies that had specifically looked at digestates from dairy manure and food waste feedstocks, also in colder climates. The few here are all from India. So no one had really looked at the US and this particular food system, this dairy-based food system. So it was an area of research that was largely untested. So I set out to go and test these residuals from this dairy manure, food waste, anaerobic digestion process as a cultivation substrate for Pleurotus austriatus to see if I could remove nutrients from these products and produce mushrooms for food. So again, this is the question: what possibilities exist for new and creative ways to use biogas residuals to export nutrients from the farms that are already struggling to manage their existing loads while also providing additional value to the farmers? And how much phosphorus could be removed if a portion of these separated solids from anaerobic farm digesters were used as a nutrient supplement for mushroom cultivation? So this strategy could be used to sequester nutrients in the form of mushroom tissues as a valuable food product, protein-rich food product. And then, so again, we introduced the mushroom farm plugin to the system, and we can capture some of that energy and heat for the process of cultivating mushrooms. And we have two new export products off the farm. One is the high-value mushroom protein food, and the other is the spent mushroom substrate. And I was also interested in how the spent mushroom substrate itself could be a useful soil amendment or compost bulking agent, or potentially a digester feedstock itself. So potentially spent mushroom substrate could be recycled back into the digester as well. But ultimately, the goal of this was to reduce the immediate land application of those nutrients in the form of those separated solids. Now, there are some other inputs that are required, typically another bulking agent. So what I'm talking about is using these separated solids from anaerobic digestion as a nutrient supplement, but I still need a primary carbon source. And so sawdust is commonly used by growers and is also frequently available on dairy farms as bedding material for cows. And any of you that have toyed around with growing mushrooms in the past know that grain is a a common supplement to supply nitrogen and phosphorus and other trace minerals to fungi so that they grow nice, big, healthy fruits. So to carry out my study and my project, I partnered with 2 commercial farms in the region. That was New Hampshire Mushroom Company and North Spore. New Hampshire Mushroom Company is in Tamworth, New Hampshire, and North Spore is in it's in Westbrook, Maine, which is right outside of Portland. So kind of these complementary farms. New Hampshire Mushroom Company is kind of out in the sticks, close to other farmlands and a lot of timber industry. So they have a steady supply of wood chips coming their way. And North Spore is really more of an urban farm. They import all their substrate, and they grow primarily off of wood pellets that are marketed as heating pellets, but these hardwood wood pellets and soy hulls is their nutrient supplement. So that's where they're supplying their mushrooms with some nitrogen and phosphorus. So they mix wood pellets and soy hulls together to form their bulk substrate, and then that's what they grow on. So I set out to test 2 different sample groups. Don't be overwhelmed by all the abbreviations up here. But group 1 is essentially, pardon me. Group 1 is essentially to determine the effect of the separated solids in a sawdust-based media. So you see R1 control up here. This is 100% sawdust. So that's my primary control. And then I experimented with different rates of supplementation. And I chose 2 different digestate materials. I chose one digestate from a farm that had absolutely no food waste. So it was entirely dairy manure, digested dairy manure, separated solids. And a second digestate, so that's digestate A. So this is primarily, exclusively of a dairy manure origin. And digestate B had the highest amount of food waste supplementation in New England of any digester I could find. So what I did was essentially to pick the 2 bookends, one without any food waste supplementation and one with the absolute most food waste supplementation, to essentially determine whether or not like all of the different separated solids available in New England might be candidates for ingredients in cultivation of the oyster mushrooms. So again, this group 1 is composed of just sawdust and the separated solid ingredients, A and B. And then group 2 was to test against North Spore's commercial recipe. And so they grow their oyster mushrooms on a 50% soy hull sawdust blend. And I wanted to see at what rate I could supplement the separated solids for soy hulls, because soy hulls are commonly sold as an animal feed. So they have kind of a high value use. They're not a waste product necessarily in and of themselves. So digestate, which doesn't necessarily have a use outside of bedding on the farm, I wanted to see at what rate I could supplement, or rather offset, North Spore's use of soy hulls for their nutrient supplements. So I tested 2 different rates of supplementation, 15% and 35%, again for the 2 different digestate materials. And this is what I found. This shows this essentially shows on the x-axis, you have the different recipes I tested, the control, the 100% sawdust being all the way on the left. And this is the yield in terms of dry grams of mushroom that was harvested off of these different test substrate recipes that I did. And I essentially trialed, I built a little mushroom house at the greenhouse at UVM, and then I had New Hampshire Mushroom Company also test those same recipes to see if it would work in a commercial setting. And I know I'm getting close on time already, so I'm gonna crank on through this. So what this shows essentially is that yes, when you add separated solids to a sawdust-based recipe, you do get an increase in mushroom yield. And then this is the group 2 group. This is the commercial recipe that North Spore uses to grow their oyster mushrooms. And then here are my different recipes. This is at 15% supplementation with material A, the 100% dairy manure, and this is the food waste. And what this shows is there's no significant difference from these two highest yielding recipes. So that's really good news. That means we can offset North Spore's use of soy hulls with about 15% of this separated solid digestate material and produce this delicious looking food product, which is safe for humans and has a high amount of protein. The phosphorus content of the mushrooms was pretty variable, which I found to be quite interesting for any biogeochemistry nerds out there. And again, they were tested for different metals to make sure that they were safe. I'm just gonna click on through this kind of fast. This slide is kind of a lot to take in, but I'm really proud of it, and it really drives the main point home. So up top here, you see just that control recipe. This is North Spore's control. And the red represents nitrogen, and the blue represents phosphorus, and the contributions to the overall substrate recipe. So soy hulls in this recipe represent the primary contribution to the substrate of both nitrogen and phosphorus, whereas the sawdust didn't add a whole lot of nitrogen or phosphorus. And what we see is we're able to harvest most of the nitrogen and phosphorus in the form of mushroom rather than what is left over in the spent mushroom substrate. So that's good news for efficiency. This is my highest yielding recipe. And again, we show greater than 100% recovery. That really is just a matter of some, it's some error in the calculations, but it's nothing to be overly, it's fine. It's essentially an error. It's a measurement error, but this is a result of 8 different replicates and an average. So what that essentially shows is that we're able to harvest a majority of the phosphorus out of this recipe as a mushroom product. So what we're left over with as a waste product is far less than what we have as mushroom food. I'm not going to hammer that again with the other food waste recipe, but again, we're left over with more overall mass in the leftover product, but that's because there was more going in the form of the digestate and soy hull additions. So the big question here is, at what scale can oyster mushroom cultivation become a realistic strategy for phosphorus recovery? And so I did a quick back of the envelope calculation and basically took New Hampshire Mushroom Company as an example, multiplied their average annual yield of mushrooms and multiplied it by 100 different farms. So if we took 100 farms the size of New Hampshire Mushroom Company, we'd be able to essentially caption about 9% of the total outflow from the first digester without food waste, and about 17% of the total digester outflow of phosphorus from the one accepting food waste. So that's pretty significant, but again, this is a matter of really scaling up to industrial level scales. And that to me makes sense because it's an industrial scale problem that we've created with industrial scale dairy. But the good news is this type of mushroom farming exists probably somewhere in the States, but the most impressive examples I found were in the Netherlands. And basically they do this at a massive scale and produce a lot of food. Now the spent mushroom substrate just represents an opportunity to capture additional value. This is at North Spore. They just chuck it all away. But there are companies out there that are selling mushroom compost as a high value amendment product. And what's nice about this is that it essentially has a higher nitrogen to phosphorus ratio. So what that means, it's potentially a better amendment for growers that are looking to increase their nitrogen content in soil without oversupplying phosphorus and risking, again, that downstream leaching. This is at New Hampshire Mushroom Company. They chuck it all out back and produce a really high quality soil that is super productive. And spent mushroom substrate has also been examined for use in pest management, bioremediation, as food for livestock and fish. So again, it's a high value product in and of itself with the right marketing, I'd say. So in conclusion, these separated solid products from anaerobic digesters accepting dairy manure and food waste can be used for commercial cultivation of Pleurotus austriatus, and optimal recipe concentrations of the material will depend on the properties of those separated solids themselves and the other ingredients used. But recipes can be optimized to recover most of the nutrient in digestate as a food product with a lower amount left over than in the residual. I'm just gonna, so I'll skip the fourth point, but then again, this could represent a potentially really high value reuse of this material and reduce the land application of nutrients nearby digesters, creating a new export market for these materials and hopefully lessening the risk of downstream eutrophication. I'm just gonna, next steps, life cycle analysis to evaluate the overall impact of mushroom cultivation, develop business models, determine economic viability, and test these materials for multiple other high-value edible and medicinal species. So take home, considerable potential exists to integrate mushroom farming with organics recycling to develop a more circular economy, but it's expected to require industrial scaling to target a significant portion of phosphorus outflow from dairy farm digesters. So there we are. We've hit on these 4 points of the food system, ways that fungi can play a role. So I'll just go back to the beginning and just what roles, again, can fungi play in a more sustainable food system? They can upcycle residual waste biomass to produce food and medicine for people and animals. They can reduce emissions and land and water use from animal agriculture. They can build soil health and reduce non-renewables fertilizer applications by using SMS to provide plant available nutrients and build up soil carbon. And they can offset synthetic pesticide use with amendments. This is not from my work, but other studies have shown, and also provide biodegradable food packaging materials. Again, not my work, but the work of others. So thank you. With that, I think we have 58 seconds for questions.
Awesome. Thank you, Brendan. Since we went a little over time, I would love to ask if we could just get 2 questions for Brendan. And then if there are more questions, Brendan, would you be willing to answer those on the chat?
Yes, absolutely.
Great.
Any questions?
Yeah. Ask a question. Can you hear me?
Yeah, Karen, go ahead.
So when you're using these agricultural byproducts, like for food mushroom cultivation, do you have to worry about like residuals from pesticides and synthetic fertilizers and things like that being present in the mushrooms?
So with regards to the fertilizers again are just nitrogen and phosphorus. So essentially the fungi are uptaking that nitrogen and phosphorus into their own tissue to produce biomass. So it's not dangerous. Metals might be a concern. Typically with these white rot fungi, they tend to produce enzymes that totally degrade organic pollutants. So pesticides, oil spills, things like that. The danger in consuming tissues grown on those types of materials is really the uptake and bioaccumulation of metals that are present. So it's a metal poisoning issue, but typically these enzymes degrade those types of organic pollutants into smaller compounds that are not harmful. So I hope that answers the question. I'm not a food safety person, so I wouldn't, I personally am not about to go eat mushrooms grown on oil, but I think, yeah, maybe there's someone else that could better answer that question with regards to the organic pollutants issue. But my impression is that basically they break them down completely, or at least into non-toxic substances, so the risk is greatly reduced. Anyone else? One more?
I'm
just curious if North Spore has been open to switching up their recipe and their cultivation practices after your research.
Yeah, I think that they have been focused on just really building their farm. They're not about to take on digestate as a material because they're really just in process of scaling their farm, and they're an urban farm. I think New Hampshire Mushroom Company would be far more willing to experiment with this material because they are more closely located to farms where these materials are produced. We get into transportation costs. That's why I suggested next steps would be a life cycle analysis, because when you start moving a lot of material that has a high water content all around the landscape, you wind up burning a lot of fuel. So proximity to digesters would be key. So that's why I kind of think of it as the mushroom farm plugin that shows up on an existing dairy farm. Take your like kind of container ship model and is able to capitalize on this steady stream of digester solids, sawdust that's being brought in for cow bedding, and also some grain that is being produced for feeding cows. So where there's close proximity to those resources, I think it makes sense. But in the case of North Spore specifically, I think that for them to use digestate, it would have to be packaged similarly to their other substrate ingredients, like soy hull and wood chips. Essentially, they come in a pelletized form, and they just add water and stir. There isn't anything to indicate that that could not be a way that digestate could then be packaged and further marketed as an amendment product. And the next graduate student that's in my old lab is working on those types of questions about really capturing value out of the digestate itself as a fertilizer product, which is preliminary work that I did as part of my thesis, but I didn't present here because it's not as fungi focused.
Cool.
Thank you. That was really awesome.
Thanks. And yeah, I'm happy if other folks want to email me directly with questions. I'm happy to follow up individually. So thank you all for your patience and interest. And yeah, I really appreciate having the opportunity to share the work with everybody. So thanks.
Thank you so much, Brendan. Let's give him some twinkly fingers. We really appreciate you. I just wanted to quickly just name that Mia Maltz is on the call. She's our lead scientist and a founder of the organization. And she might just pop in and say hi. Also, I wanted to mention about the homework for this week, where Brendan had this great assignment to create that map of energy and matter. And if you didn't complete that this week, it's okay. You can have another week to do it. And Brendan has also offered to read people's maps and give them feedback if you'd like. So I think that's pretty awesome of an opportunity. And there's instructions for that in the week 2 folder, and then I'll put them in the week 3 folder as well. So I'd love to transition now to Willoughby Arevalo, the incredible, who you all met last week, who's gonna walk us through some cultivation stuff. Welcome, Willoughby.
Hi, everybody. Can you see me? Do you see me?
Yes.
Great. So I was struggling a bit last week with the technological logistics of trying to go back and forth between sharing my screen of my PowerPoint presentation and looking like being part of the visual conversation with the camera and being able to see your faces and things like that. But I think based on watching Brendan, that means that even when I have my PowerPoint up to share the screen with you, my images, you can still see me, is that correct? Yeah? Okay, great. So that really simplifies things for me a lot. And I'll be sad that I can't see all of you while this is going on, but that will make things way easier. So I am going to share with you a presentation based on my book that is I will share it now. So, and the beginning of it is is a lot of a review from last week. So I'll kind of blaze through that a bit. And let's see if I can make it so I can see myself or see, because that would actually help me, because I'm going to be doing some demos interspersed through this. So, oh, how do I? All right. Okay. So my book, it contains, my book is tries to give an approach for people who don't necessarily have access to all the facilities that some of the other books out there, some of the other really good mushroom cultivation books that exist, kind of assume that you have access to resources and land base, such as owning land with plenty of buildings to build out a farm, and money to invest in lab equipment and things like that. My book doesn't assume that at all. It's geared towards people like me who live in a little apartment in the city, or people who live in a cabin on an island that doesn't have a ton of agricultural byproducts, or whatever, anybody really. But it's trying to break down some of the barriers, decompose some of the barriers to actually doing this mushroom cultivation work. So in the book, I give an introduction about mushrooms and human relationships, myself and my approach to mushroom cultivation, the basics of what mushrooms are, what they do in ecosystems, and what they need to survive and thrive. I kind of went over that part last time. And did folks get the overview of the cultivation process, chapter 2? Good. Hopefully folks had a chance to read that, because that really summarizes the whole book and gives a broad overview of the system I'm proposing. And it's a very short chapter, but it's really, as my editor said, the crux of the book. Then we get into workspaces, tools and equipment, sanitation and techniques to avoid contamination, starting and maintaining cultures, making and using grain spawn, fruiting substrate formulation and preparation, then bringing it outdoors to outdoor mushroom growing and mushroom gardening, harvest processing and use, and then in conclusion, substrate for thought toward further applications. The Appendix 1 gives profiles of some of the most commonly cultivated and easily cultivated species that are what we call wood lovers. I don't cover the very many of the compost lovers, but there are a couple in there. And Appendix 2 is resources. We won't get into that this time, but if you get the book, then you'll have all that. And then, so we went over this last time. This is the life cycle. At the top, we see the spores, which are released from the mature mushrooms. They germinate, seek each other out through pheromone communication, grow towards and fuse with a compatible mate, become a full set of genetic information, each cell with 2 or more distinct nuclei from the parent spores. And then the mycelium grows and amasses energy and nutrients. And then when the time is right, can produce primordia, baby fruiting bodies, which lead to mature fruiting bodies. In the mature fruiting bodies, the spores are produced on basidia and released back into the open air for the cycle to continue. Mushrooms play a wide variety of roles in ecosystems, and what we focus on with cultivation is this side of the spectrum, the mushrooms which are at least partially saprotrophic or saprobic. They decompose dead organic matter, usually in the form of lignocellulosic plant wastes. Mushrooms need air, as in oxygen, water, food, warmth, and fruiting surface to be able to grow. I'm kind of breezing through this because we went over it last week. And then this picture is giving the nitty-gritty overview, the detailed overview of what the whole cyclical process is. And if you start at the top and go from 1 to 2 to 3 to 4 to 5 to 6 and then back to 1, you end up with the full cycle. And then there are smaller cycles off the sides that can provide extra loops and expansions of the system. So we start at the top. You see a mushroom there. In this case, it's a king oyster, Pleurotus oryngii, being stabbed by a hypodermic needle. Poor mushroom, but it's gonna endure that pain and be happy for it. Because what we're doing is taking a tiny little sample of the mycelium that makes up that mushroom and squirting it into a jar of sterilized liquid culture medium, which is here. And it's got this special lid, which I'll just talk about right now. And we call it an airport lid. It's got 2 holes drilled in it. One is plugged up with silicone gasket maker, which you can get at any auto parts store. It's RTV silicone gasket maker, and it withstands the high temperatures that you find inside a pressure cannon. And the other hole is plugged up with polyfill, or pillow stuffing, if you will. Synthetic fiber fill is another name for it. And it's a little technique to fitting it in the little hole. You just get a small flat piece, and I like to kind of make a little ring with my hand and poke it through to gather all the loose ends together. And then I twist it, gathering and twisting all the loose ends to a point. So I have something shaped like a little sperm or tadpole. And then I put it tail first up in through the bottom of the hole. And then I grab it through the top, and then I tug until it's nice and snug. You want it to be pretty tight, and then I rub the underside down to kind of mat the interior fibers so they're not hanging out a whole bunch. That ended up a bit small on the inside, but not too bad. That's a workable filter. This is an air filter that allows gas exchange in and out of the vessel without letting spores and bacteria in. And then this red blob of silicone is in a self-healing injection port, abbreviated as a SHIP, or just injection port, where we can move liquid mycelium around with the needle and syringe. So the culture is started variously, but the easiest way with this system is simply to stab the mushroom, squirt that little noodle of tissue into the jar of liquid culture, let that grow, and then you can expand that to more in 1A, as you see there. You can expand that to more, become more liquid culture. And then you use the liquid culture to inoculate cooked, sterilized grain. In this case, it's rye grain. That's the most commonly used grain. And then you use a little bit of this to inoculate your fruiting substrate, which you see in 3, whether that is sawdust, straw, coffee grounds, biosolid waste from dairy farming. Woo, Brandon, that was awesome, by the way. Thank you for that. I have a friend that I really am excited to share that with. And then once that mycelium grows all through the sawdust substrate, then we move it to number 4, which is shifting the environmental conditions in which that mycelium lives. exposing it to more open air, and decreasing the temperature, increasing the ambient humidity, and introducing light if it wasn't already there. And that leads to the formation of primordia, baby mushrooms, which you see as the little bumps on the surface of number 4. And then we maintain those humid, cool, airy, light conditions, and the mushrooms develop into 5, and we harvest, as we see in 6A. We can also send that mycelium back into 6B to rest it for a little while, send it back to the primordia formation stage of number 4, and then send it to number 5, again, to fruit. And many types of mushrooms will fruit between 2 and 5 times through this cycle, through the 5, 6B, 4 cycle, before they end up going to 6C, which is the recycling of the spent mushroom substrate, or SMS. And that can be re-supplemented and re-treated, either pasteurized or sterilized, and inoculated with another mushroom species, one that prefers more broken down waste materials as its food. And of course, be used for all those other things like composting, soil amendment, biochar, potentially animal feed, etc. Woo, there it is. That's the cycle. And here's kind of what it can look like on a really DIY scale. This is how I cultivate mushrooms. So I start by making the tissue culture of a mushroom, use that little tissue sample from the inside of the needle to create liquid culture. And then I use the liquid culture to inoculate either sterilized nutrified sawdust, or the more traditional method is to use that culture, mushroom culture, to inoculate grain spawn, which then can be used to inoculate sterilized nutrified sawdust, pasteurized non-nutrified sawdust. And when I say nutrified sawdust, I mean with extra nutrients added to it, like that the solids from the what Brendan was talking about, or commonly wheat bran or coffee grounds, or okara, a waste product from tofu production, or a whole number of other nutrient supplements to add more nitrogen, especially. You can also use the grain spawn to inoculate pasteurized urban and agricultural wastes, things like straw, coffee grounds, paper products, etc. And either form of the sawdust, whether sterilized or nutrified or pasteurized, can be used before fruiting to inoculate logs and outdoor patches. And you can fruit from many of these different, there's like 5 different end substrates there that you can fruit from. We get into building, designing, finding places to work, and the tools and equipment to use. With this technique, using the airport lids and the hypodermic needles and liquid culture, you are able to avoid the aseptic transfer space. What you see here in these 2 pictures, on the left is a laminar flow hood, often simply called a flow hood, and a still air box or glove box on the right. And those are ways of allowing yourself to open the vessels of substrate or medium without a bunch of spores and bacteria falling in from the open air, which is a major vector for contamination. And using this self-healing injection port allows us to move the mycelium without having to open our vessels. So we don't need a laminar flow hood or a still air box to do a lot of our everyday mushroom cultivation things. There are certain reasons why those aseptic transfer spaces are still really, really useful. If you want to do work on agar plates, like in Petri dishes, and you can do a lot of different things with that. A lot of the development and things like that are more easily done in that setting. But these are great tools, but we don't necessarily need them to grow a whole bunch of mycelium and mushrooms. Some of the favorite lab tools for working with cultures, mushroom cultures, isolating cultures and expanding them. We see the alcohol flame in the center. You can make your own with a jar and a cotton wick and some denatured alcohol. You can, and that is used to sterilize the working ends of your other lab tools. We see the needle and syringe used to remove liquid culture. The tweezers and the scalpel are used to take mushroom tissue or mycelium from the inside of a mushroom to inoculate agar. And the inoculation loop is used to inoculate agar with spores. I showed you this in real life last week. I use my water heater closet as my incubation space. Once you get your culture started or inoculate your fruiting substrate or your grain spawn, you need a nice warm place for it to grow. It grows much better when it's in a nice warm place. Room temperature to slightly higher is ideal. So I use my water heater and furnace closet, and it's not quite as consistent as I'd like. Sometimes it gets a little high, the temperature in there, but ultimately it works just fine. And in traditional like farms or labs, larger setups, you see that usually the cultures and grain spawn are segregated from the fruiting substrates, like you see straw and sawdust bags there. But because of the security of the airport lids, you don't really need to do that. And you can use an incubation space for, in this case, that's not even that clean. Like honestly, it's really dusty in my incubation closet. And I don't think it really causes me any problems. So you want to find the existing niches that you have, unless you have the resources and space to make a dedicated incubation space, etc. You can really try to carve out these niches within your own living space. High shelves, the top of the fridge, water heater closets, things like that are great. Cabinets above the stove. These are all great spaces to grow mycelium in. And then the fruiting space is the most challenging space to create because it requires these environmental factors of reduced temperature, more or less. Some mushrooms are happy to fruit in quite warm temperatures, particularly mushrooms like pink and yellow oysters, elm oysters, warm fruiting shiitake strains, and a number of others. But usually they want a cooler temperature than the incubation space where the mycelium grew. Much higher relative humidity, usually hovering around 85%, give or take a few percent, up to even 95%, especially for the primordia formation stage. And they need lots of fresh air. As I mentioned last week, if you imagine the mycelium of an oyster mushroom or another wood-loving mushroom growing through the interior of a log, as they do, there's very little oxygen on the inside of a log, and their own metabolism produces carbon dioxide and it builds up. So for the mycelial growth, they can tolerate quite high levels of carbon dioxide. That means we can grow a culture in a jar with a 3/16 inch hole in the lid with a very tight wad of stuff in it, and it's not choking on its own carbon dioxide, even with that tiny, tiny amount of air exchange possible. But then for fruiting, that happens on the outside of the proverbial log, where it's the open air and there's way more oxygen. So that's a major fruiting trigger for them to know they're in the open air and they can fruit. And oftentimes if you are fruiting and you see your mushrooms are really stemmy, it's really common to see with oyster mushrooms, to see really long spindly stems and not very well developed caps. That's a clear sign that you need to introduce more fresh air to your fruiting space. And then gentle light. Light is a trigger for fruiting as well. And natural LED or fluorescent light is good for that. Ideally with a day to night cycle, but sometimes you can get away with 24-hour lights. I'm actually working on a time-lapse video project of myceliated stuffy sculptures fruiting in an automated fruiting chamber with my high school students. And we're gonna leave the lights on 24/7 because for the time-lapse, the lights always have to be on when the photo is taken. So they should manage okay. Fruiting spaces range from very simple and low tech, like these humidity tents, to way more sophisticated systems. Humidity tent is simply some sort of space for the mushroom to be in that is wrapped in plastic or another humidity conserving material with a little bit of airflow allowed and some sort of structure to keep the walls from touching the growing mushrooms to give some airspace. So the one on the left, it's a top view of a shiitake block sitting in a plant saucer, and around it is a rectangular piece of snow fencing with a plastic bag over the top and a couple of holes cut in the bag. And on the right, this is my personal setup that I use sometimes. It's literally just like a hanging rack from a hook and wires that I hang in a shrub outside in a shady spot in my yard. I wrap a giant plastic bag around it. It's got some holes in the top and the bottom to allow for passive gas exchange. Because carbon dioxide is heavier than oxygen, it can kind of fall out the bottom. Humid air is lighter and floats up to the top. And with this, you have to mist and humidify manually. Although in my fruiting bags, my fruiting tents you see on the right, I put some sort of moisture absorbent and wicking material, like in this case, peat moss that's soaked in the bottom. And that adds a bit of a moisture reservoir that adds some evaporative humidity. And just to also give another perspective, in places like Uganda, in rural villages where people are cultivating oyster mushrooms and others, I've heard of earthen buildings with earth floors and earth walls and probably thatched roofs with wooden racks to hang the fruiting vessels. And for humidification, they simply have burlap or other materials wicking up water out of a basin, and then it evaporates out of the burlap into the air. And that can actually do a lot to humidify a space. So it can be super low tech. It doesn't have to be super clean. At home, you can fruit in the shower. And I'm just gonna read the short passage from my book because it's a paragraph I like. So I write, the bathroom is usually the most humid room in the house, although kitchens often get quite steamy. Hang bags in the shower, but outside of the direct spray and shampoo splash zone. Mushrooms don't need shampoo, possibly with the exception of lion's mane. A few showers per day may provide sufficient humidity to support fruitings of oysters and others that don't need consistently super high humidity. And then your mushrooms get to see you naked, which may cause them to grow extra big. If showering alone doesn't provide enough humidity, you can always mist by hand too. a spare shower stall is easily converted to a dedicated fruiting chamber with the addition of racks and a humidifier. And then we get into an automated mini greenhouse. This one is not as mini as some because you can actually walk in and stand in the middle of it. But it's some sort of wire rack or shelving unit with covering, usually plastic. In this case, it's actually a glass greenhouse in this image. And humidifiers and fans for ventilation, and either lights mounted externally, or in this case, this one's actually set up outdoors and receives natural light. You can use a cool mist humidifier, like a domestic consumer model. You can also use these things, oh, do I have a picture easily, that are called ultrasonic humidifiers. I think you'll see it in one of the next pictures in one of the next slides. You can buy these really awesome ultrasonic humidifier units, and I would actually recommend sourcing them from this business called House of Hydro. They're located in Florida, and I just got some of their products. They're really well made, they're competitively priced, and they have excellent customer service. They know their stuff. You can say, hey, I'm making a mushroom fruiting chamber that's these dimensions. Which size humidifier and waterproof fans do you think I need for that? And I just asked those exact questions for my time-lapse fruiting chamber. Oh, cool. Kane, is that Kane that's showing one? Yeah, check that out. Can everybody see Kane's screen? That's a single disc ultrasonic humidifier. That one has LED lights on it, which I've heard are not so good because the lights contribute to algal growth in the reservoir. But basically you drop that thing on a little buoy into a tub of water. You can also automate the refill with toilet plumbing. Oh, there's the little buoy. Awesome. And you can have a self-filling automatic humidifier, which pumps a really high quality ultrafine mist, about 5 micron droplets into the air, which is super ideal. And then you use a fan to blow that out of there and around. And you these are ideal for fruiting rooms as well. Just look ahead. Yeah, so there is a setup with the pond fogger or ultrasonic humidifier, basically what Kane just showed, built into a trash can with a fan blowing air down into the top, and then the humid air is blowing out of holes in the sides of that trash can. You can do a little fruiting room in a closet by sheathing it with plastic, adding humidifiers, lights, and fans. The picture on the left, that little under the stairs closet fruiting space, you can see how long those stems are. That one did not have adequate CO2 disposal, aka fresh air exchange. That was my friend's closet. He didn't actually have any built-in ventilation. He would just open the door and kind of wave the door around every so often for that fresh air exchange, which wasn't always enough. So those oysters are pretty leggy, but you know, still quite edible and worth doing that way. And what you're not seeing is on the inside of the door of that closet, there were T5 fluorescent lights, little mini fluorescents that are usually used for plant propagation. And then on the right, you see a basement fruiting room with bags hanging from racks in the ceiling, buckets and bags on shelves. And that is the same one that contains this. These photos are from that same room. There's an outtake fan. You can see how it's getting gunked up by spores on the inside rim. And fluorescent lighting. And you can pump out a lot of mushrooms out of a little like base, you know, maybe 200 square foot basement like that. A greenhouse where plants live is also a pretty good space for growing mushrooms, especially under your benches where it's shady and a bit cooler. One concern though is that soil harbors trichoderma, which is great for the plants, but parasitic to mushroom mycelium. So you may have higher rates of contamination, but it's totally worthwhile. And dedicated greenhouses for mushrooms only is also totally an option, especially in the colder season. It's really good, or if you put some shade cloth so it's not overly hot in there. Another nice thing about partnering the plants and the mushrooms in the same space is they do natural gas exchange with each other. So this one is from this system where you inoculate nutrified sawdust directly in a jar with the liquid culture. And then you're able to skip the grain spawn stage. And this is the shortcut for not having to open a vessel of nutrified sawdust. When we add extra nutrients to the sawdust, it becomes much more susceptible to contamination and therefore cannot be inoculated in the open air. And it must be sterilized in a pressure canner or autoclave rather than which is lower temperature and easier to achieve and easier to scale up. So this is a pretty cool trick, kind of developed by online magic mushroom growers in the forums, and then synthesized into the sawdust format for gourmet mushrooms. And I first read about that technique, in the book Radical Mycology by Peter McCoy. And yeah, so this is a mushroom called Nameko, and I fruited this in a humidity tent in my backyard in the month of May. So contamination of the growing mushroom substrates and of the mycelium itself is a major issue that we have to contend with, which seems kind of paradoxical because mushrooms in the wild grow in soil, they grow in old wood, they're exposed to all the elements, all the biological diversity of the world. And when we grow them in culture, they kind of need to be babied to protect them from other microorganisms and other fungi. And it seems confusing, but it's partially because when we isolate them in the first place and feed them extra rich foods like sugar, water, and grain, then those are things that so many different microbes want to eat, and they will, in almost all cases, be outcompeted when they're faced with molds and bacteria. And when they are in pure culture like this, they focus all their energy on enzyme production and digestion and kind of shut down their immune system a bit, and are producing far fewer defensive chemicals to kill off competitor fungi and bacteria. And so they become a little bit weakened in that sense when they're grown in culture. But we end up doing techniques to treat our substrates, usually sterilization or pasteurization, which I kind of think like, I think of it kind of like weeding. If you prepare a very fertile substrate, like these, just as you would if you were tilling soil and adding a bunch of compost to it, aerating the soil, clearing it of existing plants, loosening it, and nutrifying it, you're going to get a ton of seeds of all sorts of plants, what we may call weeds, germinating all amongst the seeds that you plant. So the treatment process of pasteurization or sterilization or other alternatives is basically a lot like weeding. So we have a number of different vectors of contamination that can bring in contaminants or competitors. We have the air, which is a super major one, and the airport lid allows us to kind of sidestep that, which is really awesome. Otherwise, you need to do a lot of your inoculations in a clean air space, like a hood or glove box. The tools that we use, anything that touches the substrate or the mycelium can carry in contamination and needs to be sterilized, usually with a flame or by autoclaving, or sometimes with disinfectants like rubbing alcohol. The inoculum itself, if we inoculate with a culture that is already contaminated, but we don't know it, then that contamination is going to probably really outrace our mycelium in that next stage. Our bodies are super biodiverse, and our dead skin cells, the bits of stuff coming out of our mouths and noses as we speak, breathe, cough, sneeze, etc., can introduce contamination. The environment, you know, if the space is not impeccably clean and sanitized with filtered air, then that can be an issue if our containers are breached. And pests like the fungus gnats you see here, or larger animals, also bring in contamination. So here are some common contaminants that we see. On grain, we often encounter wet spot bacteria, especially when our grain is too wet. And it has that greasy kind of slimy look, and it's usually Bacillus subtilis that is the agent causing that, which is harmless to humans and ubiquitous. So it's not like a biohazard or anything, but it's going to create inhospitable conditions for your mycelium. And in the middle, in that jar, you see a blue-green mold that's most likely Trichoderma, and there are many species of Trichoderma. But Viridae is the most common one in mushroom cultivation, and it's a parasite of the mushroom mycelium, and not only a substrate competitor. So that one's particularly challenging to deal with, but as I stated last week, it's actually super beneficial for plants, and so good in our compost systems and our soils, and it's super ubiquitous. And then on the right, you see a variety of different molds. The yellow could likely be Aspergillus, the blue could be Penicillium, there could be Trichoderma, and that is grain that was inoculated by impure liquid culture. So to start and maintain cultures, we see liquid culture on the left and agar culture on the right. Agar culture is more traditional but requires that clean airspace to work in it. In most cases, however, you can inoculate jars of agar that have an airport lid on top with a drop of liquid culture. And this is an excellent way to test the purity of your liquid cultures, because in that two-dimensional plane, you can really see what grows much more easily than in the liquid raw. If you look at this, I don't know how well you can see this through the camera, but the liquid where the clouds of mycelium are not, the liquid in between the clouds of mycelium, are actually pretty clear. Whereas a cloudy culture indicates bacterial or yeasty contamination. So tissue culturing to liquid with the needle biopsy technique. So for a moment, if you can change your view to, well, actually I could just stop share and then you can see me fully, right? I see somebody else's screen though. Is everybody seeing my screen here? I don't know why I'm seeing somebody else, but that's okay. As long as everybody can see me, that's what counts. So I'm going to demonstrate this technique of doing a tissue culture to liquid culture right now. So what I've got here is a sterile syringe with a 16 gauge needle. 16 gauge is a big fat needle used for horses, like vaccinations for horses. You can get those at veterinary supply pharmacies, and they're hard to find anywhere else, basically. Needles are restricted in most countries because of drug use. IV drug use. So they can be a little trickier to find, but go to that veterinary pharmacy. And I've got a shiitake here that I'm going to clone. And this is a jar of liquid culture media that has been sterilized in the pressure canner. And you can use various recipes, but what you're going for is a 4% sugar content or less. And you can use malt extract, you can use dextrose, you can use honey, you can use corn syrup, but you can also use other things. I've used water squeezed out of giant zucchinis. I'm currently experimenting with chaga tea, which is not really sugary, but it is full of fungal polysaccharides that are probably usable by other mushroom mycelium. I'm going to try that soon. I've already used little bits of that, a little amount of that in some of my experimental media. You can also use the water that you pour off from when you cook your grains that you then use for grain spawn. And that's a little bit hardier of a medium because it's more starch-based than sugar-based. Here I have a jar of sterilized regular water, also with an airport lid on it. And normally I would not talk as I do this because I don't want to be spewing stuff out of my mouth onto my work. But for the sake of this demo, I will talk through this. So I've got a cotton ball here. I've got 70% isopropyl alcohol in a spray bottle. I've got my sterile needle and syringe. You saw it was wrapped in foil. You can sterilize these over and over in a pressure canner, 15 minutes at 15 PSI, and they won't melt. If you cook them for an hour and a half with your grains at 15 PSI, they will melt and become unusable. And then I've got a blank jar of liquid culture medium here that's been sterilized. I'm going to start by sanitizing my hands with the rubbing alcohol. So I spray that all over and I rub, rub, rub. I unwrap my syringe, but the needle cap is still on. If you're not muted, could you please mute? I'm hearing some background noise coming through. Thank you. And what I'm also gonna grab an alcohol prep pad, an alcohol swab, just like they would rub on your arm before drawing blood if you go to the doctor. And I'm gonna tear open that package. I'm going to start by using the cotton ball to sanitize the injection port, this cotton ball and the alcohol, to sanitize actually both injection ports. Scrub that silicone to make sure it's clean and get some alcohol on there to kill any spores or bacteria that are there. And I'll do the same for my liquid culture medium, and I'll come back to that. The next thing I'll do is I will hold my syringe by the cap of the needle, and then that's with my receptive hand, and then with my active hand I will squirt some alcohol on the injection port so it's actually just wet with alcohol right at the very last moment, and then quickly I'll set that down, and with my active hand grab the needle, pull it out of the cap and push it into that injection port. And then, being careful not to get my air filter wet, I'll tip that jar on its side so the tip of the needle is in the water. And I'll draw some water up into my syringe. Now I'm using a giant 60 milliliter syringe, which is great for inoculating lots of things with liquid culture. But right now, like a smaller syringe would work just fine for this process. Now I can leave it like that. It's secure. It's safe from contamination or whatever. And now what I'm going to do is I'm going to take that mushroom and I'm going to rub it all over. I'm going to re-sanitize my hands too. I'm going to rub the mushroom all over with the alcohol prep pad. Sometimes I even choose to spray directly on the mushroom with the rubbing alcohol. Well, what I'm doing is sanitizing the outside surface of the mushroom. The inside, is protected by the exterior tissues from bacteria and yeasts and other fungal spores. So theoretically, it should be a pure culture already on the inside of the mushroom. It's not always the case, but it ideally is and usually is. So imagining where you're going to stick the needle through, you want to avoid gills or any other spore-bearing tissues because you don't want spores, you just want some mycelium from the inside. And you may choose to go just through the stalk, you may choose to go through a fleshy part of the cap. In this case, I think I'll go up through the stalk and in through the top of the cap. And now I've got water, sterile water in my syringe. I'm going to give a last little spritz. I don't want to saturate this mushroom with alcohol because the alcohol will kill the mycelium that is the mushroom. And then I'll pull this out and quickly and decisively, as accurately as possible, poke through. And I'm going to look inside the needle to make sure I got a little bit of the mushroom tissue in the needle. Sometimes it takes a few tries. And then what I'll do is I'll spritz that injection port of the liquid culture media one more time, stick the needle down into the injection port, and then squirt with vigor, which does aerate the liquid because it creates a bunch of bubbles as it goes in. Through the sterilization process in the autoclave or pressure canner, you end up losing the oxygen, the dissolved oxygen in your liquid. And so you need to reintroduce some. So that's why I like to do that. Sometimes before I pull the needle out, I've already pulled it out, and this is just an example. So pretend I didn't pull the needle out. Sometimes I'll even pump the plunger of the syringe in and out a few times in the air part of the vessel to just get some air flowing in and out of the jar through that air filter to kind of get fresh air inside the jar. Then I'll pull that out. If I'm using liquid culture inside the syringe, I like to rinse them with clean water and squirt that out before setting it aside. And then that can be rewrapped and rewrapped in foil, re-sterilized later. And then I will label this with the species, the strain, the date, what medium it is, what the liquid culture medium recipe is, and if any other things that are infos I want to have, like TC stands for tissue culture, and that's usually a good one. So I'll write LE, Mycality, the name of the farm it came from. I don't know the actual strain name of this mushroom. And then the date is 2/12/20, and the medium is, I've got a shorthand code, MDYGCH, which stands for malt, dextrose, yeast, gypsum, chaga. It's a pretty fancy recipe. You don't need to go that complicated. And TC for tissue culture. And then I'll stick it in my incubation space. I also need to agitate it. So this is an important technique to develop. See how the jar is only half full? It's because we need to agitate the growing culture to aerate the liquid and break up the mycelium. There is a broken glass chunk in the bottom of the jar. I often also use marbles for the same purpose. You can also use a Teflon coated, I don't have one handy, a Teflon coated stir bar if you have a magnetic stir plate, and then that'll automate your process. But you want to do about 30 seconds a day while the mycelium is actively growing of this agitation. And you're basically swirling it as if you were swirling wine in a wine glass to get it to breathe and bring out the aromas. So if you're a wino, you're already probably good at this. And you can also do it flat on the table, which is easier for some folks. You keep it down on the table, and there's less risk of sloshing. If you do get your filter wet with the liquid inside, it's going to act as a conduit or ladder for microorganisms to get into the jar. It compromises the security of your filter. And if that happens, what you're going to want to do is get a clean syringe and suck up some of that liquid culture as quickly as possible. And transfer it to a new jar if you've got it. If you don't have a new jar of media waiting around ready to go, just stick it in the fridge and it should be able to survive there for a couple of weeks without much air. All right, now I'm gonna go back to my presentation. So, all right. Tissue culturing on agar is basically the same principle. You're taking sterile, or not sterile, but pure mushroom culture tissue, pure mycelium from the inside of the mushroom, using a scalpel or tweezers or other tool to stick it onto the agar in a petri dish. Then you wrap it up with parafilm or another barrier to keep competitors out and constrict air exchange. And mixing and pouring agar is a whole other skill to develop. And all of this work has to happen inside a clean air space or a still air space for it to not contaminate. And there's my friend Max smiling with a morel culture. What up, Max? He's not here, but shout out to Max. Grain spawn is another very important step in the process. It's like a very nutritious breakfast to lead the mushrooms into a hard day's work. So you start with raw whole grains. Rye is the most common. Millet, wheat, milo, sorghum, popcorn, and others can also be used. And there are multiple ways to prepare grain, but my favorite way is to start by rinsing it, then soaking it for 12 hours or so, draining that water, then cooking it in fresh water and bring it up to a boil and let it boil or simmer for a few minutes, just until it is al dente. You can bite through a grain without having a dry white core inside. And it's like cooked enough that you can chew it up and eat it, but it's not fully cooked because it's going to cook more in the sterilizer in your autoclave. And once it's done cooking, you want to promptly drain it of its cooking water and spread it out. I like to use this big bamboo basket as a colander, and I'll toss the grain in the air to drive off excess steam and help it not be too wet. That wet spot bacteria that we saw before is often as a result of the grain going into the jar with too much moisture on the outside of the grains. Grains harbor large numbers of bacterial endospores, which are fairly heat tolerant, and so they need to be sterilized for 90 to 120 minutes at 15 psi. And here you can see the picture in the center. I'm inoculating multiple jars of grain spawn with a single syringe full of liquid culture, just moving that needle quickly between jars with a spritz of alcohol in between each one. Some cultivators choose to flame sterilize the needle between each jar, but I just tend to work quickly and use a spritz of alcohol, and usually that works out well for me. You can see I'm working on my kitchen counter. Traditionally, the thought of doing mushroom lab work on the kitchen counter is inconceivable because of the high numbers of bacteria and spores in the home environment, and especially in the kitchen. But that liquid culture and airport lid technique really makes it possible. And on the right, you see that grain is fully covered in mycelium. Here's one I inoculated on the 7th, so 5 days ago. This is turkey tail, and it's mostly grown all through there, but there's this patch here that has not yet been myceliated, but it's growing well, and this should be ready to use within another 5 days or so. Fruiting substrate formulation and preparation. So we'll go over the containers that we use, the substrates themselves, and the ways we prepare the substrates. I know we're running out of time, so I'm going to try to zoom through this. So containers, we see filter patch bags, which have this little square built into them that allows for gas exchange. And these are made of polypropylene, which is high heat tolerant. whereas most average plastic bags that you have are polyethylene. They can withstand the heat of pasteurization, but not sterilization. And so if you are nutrifying your substrates and sterilizing them, these bags are kind of the like industry standard. Most mushrooms are grown in these bags, but you can also use glass. You can use other reusable materials. My friend Jay Schindler of Fungi for the People, he developed a system using stainless steel, perforated stainless steel boxes with a double lid with the perforations, and the perforations can be aligned or offset to be open or closed, which is pretty genius. If you ever get a chance to take one of his courses, I highly recommend it, Fungi for the People. You can also use nursery pots, as you see here. These garden giants or King Stropharia are growing from sawdust and straw cased with potting soil. In the middle bottom, you've got a laundry basket filled with wood chips that were boiled and spawned with both sawdust spawn and grain spawn. And this was done at Mush Love, an amazing farm by some amazing people in Charlotte, North Carolina. That's Nina and Charlie. And on the bottom right, that is a lion's mane that I grew in nutrified sawdust in a mason jar, inoculated with liquid culture. And at the top, there's a diagram drawn by my friend Carmen, who did those for my book. And that's a DIY air filter with a piece of toilet paper tube or other cylinder. Feed the plastic through it, fold it over, stuff some polyfill or cotton in, and then you optionally can put a piece of newsprint or other thin paper on top of that, and you've got yourself an air filter for a polyethylene bag. And substrates need to be moist but not too wet. That drawing on the upper left shows an overly saturated substrate and one that's just right with a thin film of water on the outside of each particle, thoroughly moistened within, but no excess water pooling at the bottom. As I said before, plain sawdust can be inoculated in the open air, but you need to use a lot more grain spawn than you would for nutrified sawdust because the lower nitrogen content of the substrate. And as you can see, even though I'm doing it in the open air, I'm doing it on its side. I'm opening the bag on its side. I'm not touching anything inside of the bag at all. And then I'll seal it up with that, that collar-based filter. Straw is another great substrate and other similar coarse agricultural wastes. They need to be shredded. I'm doing that by what I like to call the immersion blender technique here with a weed whacker and a barrel, and then packed into some sort of bag or cage and dunked into a bath of hot water to pasteurize it, and then inoculated. You can inoculate those in the open air with grain spawn with good technique and moderate sanitation measures. You can also do this within random invasive plants, other ag wastes, household paper waste, and things like that. On the bottom left, that's oysters fruiting from coffee grounds, which were treated only by the brewing of the coffee itself. Sterilization is 15 psi or 250 degrees Fahrenheit, 121 Celsius in a pressure canner or autoclave. Liquids, the heat penetrates quickly, so 15 to 30 minutes. Grains, an hour and a half to 4 hours, depending on your size of vessel and how loaded your unit is. Nutrified sawdust needs 2 hours of sterilization. Pasteurization is a lower temperature, 140 Fahrenheit to 170, or 60 to 77 Celsius for 1 hour or more. Now you can kind of do a fake sterilization or an approximate sterilization by pasteurizing for a really long time also. And a lot of farms are doing this. You can use external steam pumped into a vessel. As you can see on the bottom left, these two, one is the two blue barrels, one is a boiler with propane heat underneath with steam pumped through a hose to the other chamber, which is filled with substrates on racks, bagged substrates. And on the bottom middle is my fish cooler steamer with a rack and an external steam cleaning machine, and then a water bath on top. You can also ferment anaerobically, which kills off aerobic organisms. You can use hydrated lime to create a highly alkaline substrate bath that excludes most competitors for oyster mushrooms. And then there's a couple other techniques. Running out of time. You can inoculate logs and stumps with sawdust or wood plugs. You can inoculate mushroom beds with various materials inoculated with sawdust spawn. Log culture, you use either these dowel plugs or sawdust to put it into holes or grooves or wedges in the logs. You want fresh, healthy logs, not already rotten logs. Harvested ideally in the winter to spring and inoculated soon thereafter. And you can stack them up while they grow and open them up more a bit to fruit. Mushroom beds often go really well with other food crops like zucchini, beans, corn, and many others. And then you want to harvest the mushrooms before they're overly mature. You can make medicine, you can make food, you can make art. The sculptures on the left and right are ones that I did with my friend and collaborator, Carmen Rosine. The one in the middle is by Xiaojing Yan of Toronto. That's reishi on sawdust in a mold. And some of the mushrooms that are easy and commonly cultivated, like the methods I've just proposed. And you can see my family there too, Isabel and Uma. And thank you so much. I'm sorry I used up all the time with the presentation and didn't leave any time for questions. But if anybody wants to stay on and ask questions, I'm happy to stay on for as long as needed. Yeah, and you can also email me, willoughbyarevalo@gmail.com if you have more questions. And I'm also just looking at the chats.
Thank you, Willoughby. Let's give him
Yeah.
some applause. Yeah, let's stay on till 5:10. And if folks need to go now,
So can I could just
understand.
I could answer a couple of these questions. So somebody asked, O'Kane asked if micropore tape is a good substitute for the air filter, and it's an excellent, it works great, but I would not recommend doing it for liquid cultures, because if you splash onto that, there's going to be no chance of it not being a ladder. Tyvek also works. Micropore tape is really versatile though. You can kind of improvise an air filter and some air exchange in many, many types of vessels using that micropore tape, and it's super, super crucial element in the cultivation toolbox. And then the question, success rate going straight from fruit body into liquid culture, I imagine. I say it's usually pretty good. The liquid substrate, I guess I already talked about that. Cool. A lot of these questions were already answered in the chat. So if anybody has, I like the tampon. Good idea. I like to use cotton for air filters when I'm doing bags, and I'm not too concerned if the mycelium does grow through, but I would not use cotton or other natural fibers in a liquid culture jar. Yeah. Anything else? There's so many chats. I'm just kind of catching up. Any questions, anybody? It's got to be some. Ask away.
I have a question, Willoughby.
Hey, Brendan.
Hey, great presentation. Thanks a lot.
Likewise, thank you.
Why no spores in the culture at all?
That's a really great question. You can start cultures by spores, and people do, but it's really nice to know that you are working with a mated strain that is capable of producing fruiting bodies. And usually, if it's, especially if you're culturing from a cultivated mushroom, then like you can get awesome commercial mushroom productive strains by going to the grocery store and cloning mushrooms you buy there or at the farmer's market. You can also do this with wild mushrooms. The spores are more unpredictable. You don't know what if for sure if they will mate, and if they do, what that resulting genet is going to come out acting like. But you can make a spore print, squirt some water on it, and suck that up into a syringe. You can inoculate liquid culture with that. Usually I would recommend starting spore cultures on agar though, because then you can see contaminants much more easily, and then select mycelium from a healthy looking section of the plate.
I have a quick question.
Yeah.
Oh, Karen, go ahead.
Hi, Anna. Good to
Hi.
see you.
You too. Thanks for the talk. And I'm wondering about low-end temperatures for fruiting space. So like if I have a pretty big garage and maybe could keep it humid and get some airflow, like what's kind of the coldest with most of these that you've worked with?
You know, it really depends on the species and the strain. For example, with shiitake, there are so many commercial strains available, and you can get cold fruiting strains, warm fruiting strains, and wide range fruiting strains. Certain mushrooms love cold weather, like enoki, for example. It's got two nicknames that show that, like snowcap mushroom and winter mushroom. Other mushrooms like tropical pink and yellow oysters love it hot. So I would say the range of temperature, taking the extremes into account, would be 7 degrees Celsius to 30 degrees Celsius, or that's 45 to 85 Fahrenheit. But most mushrooms would prefer to be in a 10 to 24 Celsius or 50 to 75 degree Fahrenheit range for fruiting.
Great, thank you.
You're welcome. Other questions?
I had a question. Why is it that, and I don't know, maybe we don't know the answer to this, but why is it that some strains of mushrooms really don't work with this method, like morels, or do morels work and you can grow the mycelium, but it's just hard to find the substrates and the environments to get them to fruit on?
Well, morels specifically have a particular, particularly unique fruiting cycle, life cycle, where they don't fruit directly from their normal growing mycelium. They first have to form sclerotia, which are dense energy storage units. Chaga is an example of a sclerotium. Also, if you have, I'm forgetting the name of the fungus, but it's a common pathogen of garlic, and it creates these lumps on the kind of stemmy parts of garlic. That's another example of a sclerotium. They're these basically resilient long-term energy storage units of fungi, and morels develop their mushrooms from sclerotia. And so the mycelium has to encounter a nutrient poor substrate after they've already grown through nutrient rich substrate to trigger the formation of those sclerotia and the subsequent development of fruiting bodies. But
and then
yeah.
And then the sclerotia, does that that may be around in the soil for more than 1 year?
Depends.
Or is that kind of a...
With morels, I can't give you a direct, I'm not sure, but some sclerotia of some fungi can definitely endure many, many years. I don't know specifically about Morel sclerotia.
Mhm.
Thanks.
I have a question.
Hi.
We did some experiments where we were using, instead of pasteurizing the mushroom substrate, we used a probiotic.
You did what instead of pasteurizing?
We used a probiotic, like a probiotic mix of bacteria, and also we used worm compost. And they both worked equally well or better than the boiled straw that we use when we pasteurize.
Awesome.
Have you ever heard anybody else doing that?
I've definitely heard of various biological methods. Usually I've heard of Saccharomyces yeast, like ale yeast, and I've also heard of just using ambient wild anaerobic bacteria, but I've never heard of like probiotic blends. So that's pretty interesting. I imagine it is already a lot of the same species you would have developing naturally if you just soak a bunch of straw in water for a couple of weeks and let them get gnarly. But that's really cool. Was that the liquid from that probiotic, was it really smelly?
I mean, it was, it's called EM. I don't know if you've ever heard of EM.
EM?
Yeah. It's like a mix of yeast and photosynthetic bacteria and one in yogurt, that bacteria.
Lactobacillus. I have heard of this, yeah.
Yeah, so it's a pretty cool mixture, but we found it worked symbiotically with the mushrooms. It worked, it was like the best. And when we would do our trials with the substrate. And we also found that worm compost slurry worked just as well. So we were like, that was exciting because you have to buy the EM, but this worm compost slurry was
as the inoculant for the anaerobic
that's
fermentation.
A lot of time and energy, and it takes a long time. And this takes like a 5-minute soak. And then
Totally.
We're also in a.
Wait, how long are you soaking with these inoculants?
Like 5 minutes.
That's it?
Yeah, it's so much easier.
That's incredible. I would say if you're using dry straw, though, you would definitely want to soak it longer than that so it can thoroughly hydrate. But that's amazing that works in 5 minutes.
It was amazing. Yeah, especially with the worm slurry, because it was like so much faster than boiling and drying. Oh my God.
That's so cool.
Has anyone else ever tried that? Because I was thinking it'd be cool to try it with the spawn. I never tried it with we tried it with from going from spawn to mushroom, but not from like spore or so that's my next experiment, see if it works at that level as well. Because that's a lot of work, all that sterilization.
It totally is. A ton of work. Anybody ever tried that? Well, that was an awesome tidbit to share. Thank you. I'm excited to try it myself.
Yeah.
Any other questions? Well, yeah. Thanks, y'all. Have a lovely day.
Bye-bye.
And that's the end of my contribution to the course, but I may drop in the future weeks to hear what other people have to say. And I thank you all so much for doing this good work and taking the time to skill up like this and sharing what you can.
Thank you, Willoughby. It's cool. So great to listen to you.
Yeah. Again, my contact is willoughbyarevalo@gmail.com, WILLOUGHBY. arevalo @gmail.com. Feel free to get in touch and keep rocking.
Yay, thanks, Willoughby. See you all next week.